So coming back to our questions, in the samples that are collected, how would the researchers distinguish between RNA and DNA? The differences are the ones that we have just mentioned, that when they look at the nitrogenous bases, the sample that will contain thiamine will be having DNA, whereas that which contains uracil will be that for RNA. And then if they look at the structures of these nucleic acids, they will realize that DNA is double-stranded, whereas RNA is single-stranded. And then the nature of the sugars is that for DNA you have deoxyribose sugar, and then for RNA you have ribose sugar. Why is it that you can find thiamine in DNA and you also find uracil in RNA? The point here is that thiamine is more stable compared to uracil, and therefore, if you find it in DNA, it prevents mutations, okay, to some extent. Whereas uracil, which you find in RNA, is actually short-lived and does not require the same long-term stability that you actually need in the DNA. So that's the benefit of having thiamine in the DNA and not having uracil in the DNA. The uracil in RNA is not stable for a long period of time, and that means that the RNA is prone to mutations, that is changes in the nature of the genetic material, that is genes or chromosomes. So the third question was that which of the nucleic acids actually degrades faster at high temperatures and why? You should expect then that the RNA degrades faster because it is just a single-stranded molecule compared to the DNA, which is double-stranded. But again, the sugar that we have in RNA, which is ribose, is more reactive than the deoxyribose sugar that we find in the DNA. Okay, so that's why you would expect that RNA should be degrading faster because of it being single-stranded and also having a reactive sugar. That means that it can react and then get changes, get structure changes. The fourth question is that why is DNA stable compared to RNA? Okay, because RNA is actually transient. And the answers are that DNA is double-stranded and that provides it with protection. And the fact that it is double-stranded means that the strands are actually connected via hydrogen bonds that also contribute to their stability. The fact that RNA is single-stranded means that it is prone to even be broken down by enzymes. In the questions, we saw that they used labeled radioactive substance. When you have labeled radioactive substance to use in research, you are interested in observing how that labeled molecule moves in living organisms. Like you can perhaps be able to explain how a particular biological process happens. And that's why when you have labeled molecules of RNA or even DNA, you can be able to see what movements happen, and then you can be able to note the different roles that they play in a process. For example, protein synthesis. If you have replacement of a particular nitrogenous base with another, that alters the nature of protein that will actually be synthesized, because it is this arrangement of the nitrogenous bases that determine the nature of amino acids and eventually the nature of proteins that will be synthesized. So any change will definitely result into changes in the nature of proteins that will be synthesized. In this case, a change in nitrogenous base, where you have a replacement, it is what you can term as point mutations. But of course, point mutations can be by deletion, by replacement, or by transfer. Anything can happen, and that alters everything. Okay, so in this case, if you have a point mutation, that is a replacement of one nitrogenous base with another, then in some cases you can have a disease coming up. In some cases, you can have it happening, but you don't have a health problem emerging out of it. So you can have a silent mutation when you have no effect manifesting, so no impact. Sometimes you can have a point mutation that results into a health problem. Okay, but sometimes you can have a change that results into a change in the particular amino acid that will be synthesized. So a number of things can happen. Some of the changes can result into stoppage of protein synthesis. If protein synthesis is going on, and then when that code is reached, it signals that protein synthesis should stop. So a number of possibilities can happen when you have a replacement. So what precautions should be taken when dealing with issues to do with employing genetic research techniques? When you alter the genetic makeup of, for example, plants or even animals, they are likely to have long-term impacts. This impact may not be predicted. So it means that when you're carrying out research, you need to be very careful and take the necessary precautions. What you would want to have as one of the precautions is that the changes in the genetic makeup is targeted. That is, you only focus on what you want to achieve, such that you don't damage the other sections of the genetic material. So that is what we term as precise gene editing techniques or tools. The other thing is that before some of these modifications are introduced to the population for use, for example, in this case, the issue was to make sure that you have disease-resistant plants. It means that before these are introduced to the population, you must have carried out long-term studies to understand what is the long-term impact of having strains that have been modified through genetic modifications. Okay, and then lastly, if you have organisms that are using RNA as their main machinery for synthesis or for survival, and then we have organisms that are DNA-based organisms that for them they use DNA, you will notice the difference between these two groups of organisms. Those that use RNA mainly, you will realize that for them they use RNA-dependent RNA polymerase enzyme to be able to replicate. Okay, and this may not be very efficient in doing what you call proofreading. That means that errors are likely to happen in these organisms because of the fact that the enzyme RNA polymerase does not have the ability to check and see if there are errors, and therefore mutations are likely to happen in RNA organisms. Okay, but we have some RNA organisms that actually have what you call reverse transcriptase enzyme, for example, the HIV. This enzyme means that it can turn RNA into DNA, and therefore the DNA can drive its activities. So therefore, on the other hand, you find out that the DNA-based organisms are the ones that have an advantage because they can do this proofreading and take and make sure that you don't have a lot of mutations. That marks the end. Thank you.